Hey guys, in this video we'll be talking about how to determine the primary structure of proteins. And the primary structure constitutes both the it's basically the sequence of amino acids that make up the polypeptides of a protein. So let's start. The primary structure comprises the identification of constituent amino acids with regard to their quality, their quantity, and their sequence in a protein structure. Quantity, how many of them we have, and the sequence and the quality have to do with what or which one they are. Now this is a requirement and can come in an MCQ. A pure sample of a protein or a polypeptide is essential for this determination, which involves a total of 3 steps. And if you have an SCQ in which they ask you to write down how a biochemist or you would determine the primary structure of a protein, firstly you're going to have to write 3 primary steps before giving the details. First is you need to determine the amino acid composition. That is which types of amino acids are present and how many of each amino acid is present. That is the quantity and the quality. Number 2, you'll have to degrade the protein or the polypeptide into smaller fragments. And the reason for this will become clear sooner. And then you determine the sequence of the amino acid. And you need to do the second step because the reagents that we use in the third step don't quite work well when we use large fragments. So we'll start with the degradation. My apologies, with the determination of amino acid composition in a protein. This is an important word. Determination of amino acid composition in a protein. In the first step, you don't write sequence, you write composition. Because even though we know which amino acids are present and how many of them, we don't know where they are present relative to each other. The protein or the polypeptide is completely hydrolyzed. What that means is that all of the constituent amino acids, all of the residue amino acids, they're sort of cleaved, they're liberated. To liberate the amino acids which are quantitatively estimated. This is again an important word, that the protein or the polypeptide is first of all completely hydrolyzed. Complete hydrolysis of a protein is necessary for the determination of amino acid composition. And after complete hydrolysis of a protein has been achieved, the next step is quantitative estimation. Estimation can be of 2 types, qualitative and quantitative. This is quantitative estimation. And this complete hydrolysis, which is a requirement for the determination of amino acid composition, can be carried out in 3 ways. It can be an acidic hydrolysis, an alkaline treatment, or enzymatic. In general, enzymatic hydrolysis gives only partial hydrolysis. Right, and now they've given an example of enzymatic hydrolysis. These examples are important. They might even ask you what pronase is. And a pronase is a mixture, so not just one. It's a mixture of non-specific proteolytic enzymes that cause the complete hydrolysis of proteins. Right? So generally speaking, if you were to use enzymatic hydrolysis, you would not be able to achieve the complete hydrolysis necessary for you to carry out quantitative estimation of amino acid composition. However, if you were to use pronase mixture, which is basically a mixture of non-specific, meaning these enzymes, they don't cleave the peptide bond between specific amino acids, but is rather non-specific. It sees a peptide bond regardless of the amino acids on either side, it will cleave it. It's a mixture, so not just one non-specific enzyme, but multiple types of it. And proteolytic, meaning it's responsible for peptide bond breakage. And why we have to mention non-specific will become very clear, because in another step we'll have to use specific proteolytic enzymes. So you need to really remember what pronase is. This is important. Now if we talk about the separation and then the estimation of the amino acids, we have successfully liberated all of the amino acids. Now we have to separate the various amino acids from each other, and then we have to estimate them. And we need chromatographic techniques for this, in which amino acids are separated on the basis of their different RF factors. Right? You don't have to go into detail in this particular question, but you need to know what chromatography is. Degradation of protein into smaller fragments. So assuming you have carried out the first step successfully, you have a chromatogram and you will compare the RF factors of each, right? You will compare the RF factors of each with known RF factors, and you will know that this amino acid and this amino acid and that amino acid are present. And you will have an idea of just how many of them, right? But now you need to know what the actual is. But now you want to know where the actual amino acids are located relative to each other. And as previously mentioned, the reagents needed for the last step, they don't quite work as well when dealing with larger protein polypeptides. So we need to break them up first. But that can itself be a problem. You need to listen carefully. Protein is a large molecule, and it is sometimes composed of individual polypeptide chains. And before you break a single polypeptide into smaller peptide units, you actually need to separate the individual polypeptides. Now, an important thing is, because we have to do sequence determination, we need to actually know how many we need to know which polypeptide was which one, where it was located. More than that, we need to know how many polypeptides were there. Even if we have the reagent that sort of isolates the polypeptides, we still need to know where they are located. And we also need to know the sequence of the peptides of a single polypeptide, because even if we were to figure out the sequence of individual peptide units, how are they arranged relative to each other? That is a bit of a difficulty, isn't it? We will be talking about this in a moment. How do you liberate the polypeptides? So you need to use reagents for that. In general, the different polypeptides that make up the quaternary structure of a protein, they are linked via non-covalent bonds, with the exception of certain disulfide linkages, which we will talk about in a while. So what are the reagents of choice? We may use urea, and that is the one that is easy to remember, but I would still recommend you remember this as well. Urea, or guanidine hydrochloride. Guanidine hydrochloride. It disrupts the non-covalent bonds and dissociates the protein. We have protein made up of 4 polypeptides, and so that single protein will dissociate into 4 polypeptide units. For cleaving the disulfide linkages, these two won't work. We need treatment with performic acid, right? And without it, the disulfide linkages will remain intact, because these are again a type of covalent bond which is different from a peptide bond, but still much more stronger than non-covalent bonds. Right, so we have liberated the polypeptides, and if we were to sort of break them up, it'd be really confusing, right? Because we might assume, for example, that there were just 2 polypeptides, and based on that logic, with all of the clues we have, we may come up with a wrong orientation, a wrong sequence. So you need to know how many there were. And for the sake of liberation of the polypeptides, we used 3 reagents: urea, or guanidine hydrochloride with urea, or guanidine hydrochloride, and for the breakage of the disulfide, we used performic acid. How do we identify how many polypeptide chains that make up a certain protein? We need another reagent, we need another protein, and that is dansyl chloride. So guanidine hydrochloride and dansyl chloride. Now this protein, it specifically binds with N-terminal amino acids to form dansyl polypeptides, and these polypeptides on hydrolysis yield N-terminal dansyl amino acid. And the number of dansyl amino acids produced is equivalent to the number of polypeptide chains in a protein, right? So we have a protein, let's say this is 1. I could simply use a register for this kind of shit. We have 4 polypeptides that make up a single protein, right? And it was kind of like this, but we have separated these into 4 polypeptide chains that are dissociated, or we have dissociated a protein with a quaternary structure into its polypeptide subunits. And for that we have used 3 kinds of reagents: urea or guanidine hydrochloride for the sake of breaking any non-covalent linkage such as a hydrogen bond, and we used performic acid to break the occasional disulfide linkages. Now we need to know how many polypeptide chains made the rest of these, and for that what we use something known as dansyl chloride, because this sort of comes over there and this is the N end, right? This is the carb C end, and it forms this N-terminal dansyl amino acid. And when we degrade the first amino acid, and there are plenty of ways of doing this, and when we carry out chromatography to figure out how many dansyl amino acids there are, and there are ways of doing this, the number of dansyl amino acids will be equivalent to the number of initial polypeptides. I'll read this again, this is a bit confusing. The number of polypeptide chains can be identified by treatment of the protein with dansyl chloride. The name of the reagents are very important. And this dansyl chloride specifically binds with N-terminal amino acids. Which amino acids? The ones on the N-terminal of a polypeptide chain, and they form dansyl polypeptides. And when we hydrolyze these polypeptides, you get N-terminal dansyl amino acids, and the number of these is equivalent to the number of polypeptide chains in a protein. Now we know we have the polypeptides isolated, their number accounted for. Now you need to fragment the polypeptides, and you can use enzymatic or chemical methods. You can use either of these methods. The only thing that needs to be accounted is that these methods, they need to be specific. You need to know which bond is being broken up, or the bond between which amino acids. You can use a couple of specific proteolytic enzymes, and the one that is most commonly used is trypsin, which only breaks those peptide bonds which have either a lysine or an arginine on the carbonyl side of the peptide linkage. Again, you need to remember this, they might ask you in a short answer question. Chemical cleavage, you use cyanogen bromide, and this specifically splits peptide bonds which have the amino acid methionine on the carbonyl side. And then how do you do the actual determination of the amino acid sequence? Right? Now you have the peptides. What you use is 2 methods, the Sanger method or the Edman method. The Edman method is by far the more common and the more used. In Sanger's method, the reagent that was used was 1-fluoro-2,4-dinitrobenzene. And what this does, it that this reagent specifically binds again with the N-terminal amino acid, and what is formed is basically a dinitrophenyl derivative of the peptide. And when you hydrolyze this, this on hydrolysis yields DNP amino acid, right, which is dinitrophenyl. You can just remember this, and free amino acids from the rest of the peptide chain, and you can identify this amino acid via chromatography. So with this method, you can only figure out the first amino acid. That is the limitation of this. Even so, this can be pretty useful. Edmonds reagent isn't quite so limited. The reagent used is not 2,4-dinitro, 1-fluoro-2,4-dinitrobenzene. The reagent is phenyl isothiocyanate, and it reacts with the N-terminal amino acid to form a phenyl thiocarbamyl derivative, which on treatment with a mild acid known as phenyl thiohydantoin, you get this cyclic compound liberated, and you can do this successively. One will be cleaved because of the second reagent, and you will be left with the sequence with the exception of the first one. Another amino acid will now be on the terminal end, which will sequentially be broken apart, and one by one you can identify each of the amino acids via chromatography. So this time you need to know 2 reagents. Important, so I'll write it down for you. The first reagent is phenyl actually. Phenyl, you need to remember this. Phenyl, and iso means basically has to do with isomers and such. Phenyl isothiocyanate. This is the first reagent used in Edmonds, which is basically Edmonds reagent. If they ask you what is the Edmonds reagent, you need to give the chemical name, which is phenyl isothiocyanate. You need not confuse this with the second reagent, phenyl isothiocyanate. And if they ask you how this works by binding with the N-terminal amino acid of the peptide, right? So peptide has the N-terminal amino acid, and this reagent will bind with it. And what will be formed? Phenyl thio, and that much is same, except it's phenyl thiocarbamyl derivative. This is what you need to remember as well when you're writing this down. And then what do? Is this the end of it? Previously, we didn't talk about any particular method of hydrolysis, and we mentioned that all of the peptide bonds will be broken. This time we have another solution. We hydrolyze with a specific reagent, a mild acid. That is, and what is the mild acid? It is phenyl. Again, the phenyl part ought to be easy enough to remember. Phenyl, then we have the T part, then we have the H part. Thio again is easy. This is the next part that is a bit different. Hydantoin. What does this do? It liberates a single amino acid, which you term as the PTH amino acid. This is a cyclic compound, and because just one is liberated, you can identify via chromatography. And this is advantageous since a peptide can be sequentially degraded. This is a term, even though you need to write this in your own words for your own sake, you need to remember these keywords which make your explanation all that more easy to get for your examiner. Sequentially degraded. Liberating N-terminal amino acids one after another, which can be identified. And why is that? Because the peptide as a whole is not hydrolyzed but only releases PTH amino acid. Hopefully you guys found this to be helpful. If you did, please like, subscribe, and leave a comment.